Here are some instructions that I would request all of the audience to adhere to while the session progresses. All participants are requested to keep their mic muted throughout the session. Please post your queries and comments in the chat box. The resource person will address them at the end of the session. Ensure that you fill up the feedback form, which will be provided by the end of the session. This would be considered mandatory for issuing e-certificate.
We are moving on to the second session titled Innovations in Assisted Reproductive Technologies, Impact on Contemporary Clinical Practice and Future Advances. I welcome all the participants to the session and I invite Ms. Shabana Said, Head of Embryology and IVF Clinic Hoskin Group of IVF Clinics Norway to conduct the session. Over to you, ma'am.
Hi, good morning. And I'm happy to see that there's a lot of you in the morning waiting to listen to the next session. And thanks again to the Department of Biotechnology and the staff for giving me this opportunity to explain to you what we do in our daily life. Last week, I'll just share my screen. Can you see the screen?
Yes, ma'am.
Yes, ma'am.
Okay. So last week, I was able to explain to you what ART or Assisted Reproductive Technology basically means, what we do in our daily life, and what it actually entails. This week, we are going a bit more further. We're going to talk about innovations in ART, how are innovations introduced, and how does it actually impact, and what are the future advances. I think as biotechnologist and molecular biologist and as biologist, you'll be interested to know that this is a rapidly developing field and a lot of new technologies and innovations come every day. If you remember, I mentioned last time that the first IVF baby was born in 1978. So within a period of a little bit over 40 years, you can see that the IVF, the whole industry has changed a lot. From just being simple methods to make a couple pregnant, it now includes everything from artificial intelligence to automation and high-tech laboratories. So today I'm going to discuss about when should a new technology or an innovation be introduced in our clinical setup. Because when you start working, you will see that there's a new development that's happening all the way. Like there's something new coming out. And then you're like, okay, should I have it? Should we introduce it into our clinical practice? How do you validate these innovations? Because these innovations may work very well in another clinical setup. The studies might be conducted in a different country. So how do you know and how can you validate them in your own setup? And you also have to make sure that you're creating innovations without actually cutting corners. And that's about balancing both safety and efficacy. And finally, when you have finally done all the first three things, then how do you really implement it? So that's what the talk is going to be about. So like I mentioned, we started off with these small TV screens with a computer connected, but now we have automated systems. The dishes have changed. The way we practice embryology has also changed. So there comes the provocative question. Should we or should we not introduce a new technique into our system? So there are these three questions that always have to be asked, whether you work in embryology or in any field connected to biology. Why do we really need a new technology? Do we need a new technology? Does it improve our outcome? The outcome in this case is live birth of a healthy baby. If you think that you need a new technology, how do you choose the right one? Because you see in the market, you have a lot. For example, if you guys are going to buy a new iPhone or a new telephone, then you will think about, there's a lot of new ones, which one would be the right one for me? And then once you have decided the right one, how do you implement this into your daily practice? even though this article, it's an open access article, is related to IVF laboratory, I really recommend you to read it, because when you are going to start working in different fields of biology, you will come across these things like all these technologies are highly expensive. So you have to really make a decision what you want to do. So there comes this first part. You create a hypothesis, you do research about it, you study for many years about the basic physiology, in this case of embryo development. And once you're sure your hypothesis is correct, then you have to test it out in animal models. For the humans, you normally test it out in mouse models and then move on to bovine, or the cow eggs and the pig. Why the mouse models are decided? It's because you have a large number of eggs produced. They have a shorter period of ovulation, so you can within a month stimulate the mouse a lot of times to get a lot of eggs. When you move on to large animals like the bovine and the pig, these are much more bigger. You don't really need a microscope to see a bovine egg. It's quite big. So you test it out in them. And if your hypothesis is proved there, then you start testing out in human embryos that are donated to research. And this is important because when you test it out on not so good quality embryos and they're donated to research, then you can test it out in small scale single clinic studies. And if your hypothesis still gets confirmed there, then you have to test it out in large multi-site clinical studies. And when all of this is done, then you still have to check whether it's clinically effective and whether it's cost effective. Like the money that you put in, is it worth it? So every procedure that's involving its application to the human body should be defined as experimental until you have adequate scientific evidence that's provided both with regards to safety as well as efficacy. So for example, in the UK, for IVF treatments or anything related to assisted reproduction, they have this traffic light method. So for example, they have the 3 colors, green, amber, and red. If it's a green one, then it shows that a large number of randomized controlled trials have been done, and it has been completely proven with regards to efficacy as well as safety. So for example, you remember I showed you the video of ICSI, or where you injected a sperm into the egg. That's proven beyond doubt. Then you have the amber color ones, or slightly orangish. They're the ones there's not yet clear clinical evidence, so you can't really provide it to the couples. You can inform them about the options. You can give it, but you cannot charge the patient any money for that. Then you have the red ones where there's no evidence to show that they improve the chances of having a baby. So these red light signals helps the patients to decide, or these traffic light signals helps the patients to literally decide. So all the different techniques that are offered, including pre-implantation genetic diagnosis, you can see in this case as a traffic light rating of red. So clinics are not recommended to provide these treatments to couples. So when you have a new technology, it can also be a new innovation. You're looking at a new biomarker. Biomarkers are normally those markers that will provide you, that will give you an insight into whether it's effective or not. For example, say you have to check the egg, the amount of eggs that your body still has. Then you have a biomarker called AMH, or anti-Mullerian hormone. So when you do the measurement of AMH in any teenager or anybody, and if you get a low value, it will give an indication that you don't have so much of eggs in your body. If you remember, I said a baby girl is born with all the eggs that she's ever going to have in her life. So then that literally means that this young girl should not delay her childbirth, or she should not wait a lot of time to have a baby. If she wants to do her career, then she should have the option of socially freezing her eggs. So one of the most important correlation is to see whether the biomarker has any correlation with the outcome. Then you have to check about the safety, the efficacy, and practical utility. It should be something that should be easily measurable. For in the case of AMH, the blood is where you measure it. Then if it is something that can be quantified, then you quantify it. So all these things are to be considered when any innovation is introduced. Now, a really easy example is the coronavirus vaccine. So you know that we have to create innovations without actually cutting corners. There have been this criticism that the coronavirus vaccine came into the market very fast, and were they really effective clinically, and were they safe? So I thought this was the best example to show you. So if you think about it, by January 2020, the scientists were able to unlock and decipher the coronavirus secrets. How did they manage to do it in a very short period of time? Because they had the full information from the Human Genome Project. So if you remember, the Human Genome Project took almost 13 years to complete. But since we had this full information, it was much easier to decipher the coronavirus structure. So in this one, you can see the red one on top, that is the coronavirus spike protein, and that attaches to the angiotensin converting enzyme 2 receptor on the human cell. And that's how it enters into the human cell and causes an infection. So once the structure is deciphered, it didn't take them 13 years, it took them just a few days. Then the next thing was how to develop a vaccine strategy. So the best way to do a vaccine strategy is basically to find or to inject a non-functional viral mRNA strand, a partial strand, and that can be delivered. So that will translate copies of proteins, and the immune system will recognize as foreign. So it will develop antibodies against that particular virus. So when the actual coronavirus tries to penetrate into the cell, it will say, okay, this is foreign, I have antibodies against it, let me attack it. So this vaccine development strategy was actually cut by 4 years. Why was it cut? Was it cutting corners? No, because we had a lot of information from SARS virus, MERS virus infections, and the viral structures were much known. So, but then you have to inject, you said that we could inject viral mRNA that was inactive or non-functional. So how could you inject it? So the next thing was to find something that the virus could be, or the viral mRNA could be injected with. Because if you inject the viral mRNA as such, then it will biodegrade and it will just become ineffective and it will not create an immune response. So you need a delivery system. And to develop the delivery system, it didn't take the scientists much time because there's always been studies going on to develop an HIV vaccine. So when you need an HIV vaccine, there's the same thing is there about the delivery system. And they found out that the same delivery system that was used to develop a HIV vaccine could be used even in the coronavirus thing. So these are called payloads, and they're actually tiny fat bubbles or lipid nanoparticles. So it's always interesting to see that when you have one branch of biology, right, because you're producing vaccines, then you also take the help of some other branch of science. So I think that's one of the most interesting things about science, that it's all interconnected. So in this case, we're using nanotechnology, using tiny fat bubbles to deliver this viral strand. So you can say it was cut short by 4 years. And once this was done, then you needed volunteers to test it. The volunteer recruitment normally takes around 2 to 3 years. But in the coronavirus strategy, recruiting volunteers were not so difficult because there was a pandemic that was going on. There were a lot of people who were interested in not getting the disease and of course not dying. So they were recruited at a faster rate. It just took them 2 weeks to recruit enough volunteers. And we were also getting fast results because it was a pandemic. Globally, you could test it out in large multi-country trials. So then came the fact about approvals, guidelines, documentation, ethics, and safety. So even though this coronavirus vaccine production happened quite fast. It did not cut corners. They were looking at the two magic factors, that was safety and efficacy. So there's also this thing about how can you make sure that any innovation that comes in has been properly tested. So I thought, again, the coronavirus is a good example, because you're comparing vaccine development throughout history. You can see in the 1900s, early 1900s, it took a lot of time for animal studies, the human trials, rollout, and so on. But with advanced technologies, it is much more easier. And once the Human Genome Project came, then vaccine rollout has been much faster. So if you really think about it, if the swine influenza vaccine, or the swine flu as it's called, it came quite fast. So any innovation can be introduced as long as it doesn't cut corners and its safety and efficacy has been proved. What about the IVF labs? A large number of new technologies have been introduced. IVF is the new booming market, right? Because everybody wants children. People are willing to put in money. So a lot of new things comes into the market. So like what I said last week, we have this time-lapse imaging that's completely changed the way IVF or embryologists check and annotate embryos. So you can see the picture of a 4-cell embryo here. This was how an embryologist annotated. You took out the dish with the embryos, checked it under large inverted microscopes with 40 to 100x, 100 times magnification, and then just did less than a minute variation, or you have to do it as fast as you can. So you didn't get much information. But when time-lapse came, you could actually get a lot of information regarding what actually happens. It's like a 24-hour surveillance camera that literally tells you what the embryo did when you were sleeping. So you get more information about the morphology of the embryo, the kinetics, because like what I mentioned last time, what is more important is how synchronized the divisions are and at what time divisions happen. So timing and synchrony are quite important. So when you were previously a blastocyst, that's a time just before it implants into the uterus. It's a period of 120 hours after it's fertilized. You would be probably taking out the embryos 4 or 5 times a day. But now with all this information, you're getting a video of around 120 hours. So a lot of biomarkers can be created along with this. Another thing is about omics. You probably have learned about metabolomics and proteomics. So what they do is you have these culture dishes where the embryos are cultured. The embryos will release a lot of metabolites and proteins into the culture system. And then you can measure these metabolite concentrations. And then you can say, okay, in this this embryo that literally became a baby had so much quantity of metabolite that was produced. And it can be anything from ammonia in the culture well. That's what you call as a spent media. Or it can also be the amount of like oxygen that was consumed by the embryo. So metabolomics and proteomics were together added to this morphology to increase the chances of success. So when you have these two embryos, the embryo number 1 and 2, do you see any difference between them? They are both 4-cell embryos. They look exactly the same. But when you have the time-lapse system, if you look at this first embryo, the normal cell division pattern or any cleavage pattern is as shown below, because you have a single cell, that's an egg. Once it's fertilized, it's a zygote. Then it can divide into 2. And then each of the cell divides into 2. Why does it divide into 2? Because we have a bipolar spindle. And when we have a bipolar spindle, any division of a cell to more than 2 is actually a cleavage error. So in this case, you can see the embryo did everything exactly as it wants, whereas in this embryo number 2, you can see it divides from 1 to 4. So it has a cleavage error. Otherwise, you would not see any difference between these two embryos. What's the issue with having a cleavage error or a fail in the or an error in the division? You can see this big study from a Spanish group where they say that you have transferred almost 1,700 embryos. You had a live birth rate of less than 1% in these kind of embryos. So you can see that this can be used as a deselection marker, and you can say that all the embryos that have these kind of cleavage errors should not be transferred. So time-lapse has literally improved, or it's a good way of doing an embryo selection. What about different embryos? You can see that I talked about morphokinetics, which means that you have a different kind of embryos. Your aim should be to have one single healthy baby at a time. When you have multiple pregnancies, twin pregnancies, they literally can affect the mother. You have a lot of preterm deliveries, low birth weight for the child, premature births, and countries like in the outside where the public system pays for your health care, then it creates a lot of stress on the healthcare system. So then it's important to select the right embryo. So say, for example, in this case, you have let me play the video again. You have a lot of embryos that look exactly similar. How do you know which embryo is the best one? Excuse me. So then you'd look at the timing and the synchrony to select that best embryo. How does it help? It helps basically to reduce the time to pregnancy. You're able to see the cleavage errors. You're able to see whether there's chromosomal abnormalities, but you're also able to select the right one that did everything at exactly the right time. So then with one stimulation, she is able to have many pregnancies, but one at a time. So that's about time lapse. What about other automation? Basically, we are moving towards more robotic IVF labs. It's good because it allows consistency. You do not have operator variability. One of the most important things in the IVF lab is you have to be systematic, and it has to be like you have to deliver results each time. So consistency is important. Consistency between operators, right? Because my results should be equally good as my colleagues' results. It's also important that maybe I'm having a bad day today, so my results for today may not be as good as my results for tomorrow. So to avoid all that and the reduction of errors, automation or robotics have come into the market. So what about microfluidics? I assume that you have learned a bit about microfluidics. In IVF, they're called IVF on a chip, or they're also called as organ on a chip. Microfluidics are in its initial stages in IVF. These are actually PDMS chips. They're very small. They are polydimethylsiloxane chips. They are biocompatible, they're transparent, they are gas permeable, and they are elastomeric. So basically what you're trying to do is have a streamlined movement or a laminar flow where all the different fluids, or like for the media, they all travel, or you can say they all flow in the same direction, but as separate streams. So you can see in this one, you can see that there are separate streams for media movement. And these are gas permeable. So in IVF, all the equipments you are trying to have, or you're trying to have them as close to your body. So you give importance to the oxygen levels, the carbon dioxide levels. So these are gas permeable and they're very helpful. They also have the Braille pumping system with tiny electric piezo actuators. So you have a streamlined movement, you have gas permeable, and you are trying to have the whole lab in a chip. How has it been properly used? It has been used in sperm manipulation. You have an inlet for the semen sample, you have an inlet for the media, and then you select the sperms that are highly motile by traveling them to a new channel. They've also been used in fertilization and embryo culture and cryopreservation, not yet in the human beings, but studies are being conducted in the pig. So this will allow consistency. Basically, you put the sperm and the egg at one end, and then you collect the embryos at the other end. What about artificial intelligence? Artificial intelligence, or AI, has been recently one of the most hot topics in IVF. How can we use it to develop predictive models? Predictive models for euploidy or aneuploidy testing, for pregnancy, for implantation and live birth. So ART, it is basically, or AI is basically to support clinical decisions. So now I said that we have a time-lapse videos, but still the decision is the embryologist, right? You are the one deciding which is the best embryo to put back. So how does AI work? This is the simplest example of an AI work. You give the computer the same instruction every day. How are you? And then the computer should be able to give you the answer, I am fine. So it's just the same way. This artificial intelligence tool called Ivy from Australia, excuse me, you feed the neural network thousands of embryo time-lapse videos. More the videos you feed it, and you tell this neural network, this particular embryo gave me a live birth. So afterwards, it will be able to recognize which embryos will give a viable pregnancy. So this has been tested out in many clinics, and they say that if an embryo looks like this, then there's a 45% chance that it will be a baby. When the embryo looks like this, it will have a 75% chance. So this was one of the most, the first Ivy was one of the first artificial intelligence tool that were used to predict the chances of a viable pregnancy. You also have something called the Eos System. That's the Early Embryo Viability Assessment. So this is now readily available and widely used. So basically, you allow the embryos to be cultured. Using dark field microscopy, the embryos will be graded by the machine, and then it will give you a score of 1 to 5. So score of 1 gives you a prediction that there's a 76% chance of it becoming a good quality blastocyst. 2 has a 54% chance. So you can see that if you have embryos like this, you are able to see, and this is based on more than 100 variables that the human eye cannot see. So this is one of the automated, or you can say the AI systems that's already in the market. You also have something called Eda Score, and this is an intelligent data analysis for embryo evaluation. Again, the different embryos get scores from 1 to 10. 10 is the perfect embryo, 9.9 is a perfect embryo, and 1 is the embryo that's not at all perfect. So to the human eye, some of these embryos might look really good, but when the Eda score might be quite low, then you have this extra decision-making tool that tells you whether to put this embryo back or not. So what does AI help in assisted reproduction? You have this whole thing about culturing the embryos. You can extract data from both the inner cell mass and also from the trophectoderm. If you remember this inner cell mass, that's the one that gives you the baby proper, and this trophectoderm on the outside will be the placenta. You can also use the spent media from the dishes, do an immunoassay and a pre-amplification and immunoassay, and then you can detect whether the protein levels. So you combine these two, feed them into the AI networks or the artificial neural networks, and then based on a number of criteria, you just have this option of saying, okay, if this morphology is like this and the protein levels are like this, then you have a chance of having a live birth. So these are in its initial stages, but being tested out in different parts of the world. So you're combining your culture along with artificial intelligence, and even though it's in its initial stages, a large number of algorithms have been developed that will help maximize studies. And I think AI is going to be the future. So we have now talked about basic innovations that are directly visible. Another thing is pre-implantation genetic testing. So how do you test? So, you know, when you get pregnant, you can test it by amniocentesis or chorionic or CVS, chorionic villus sampling. but those have the risk of early abortions. So this is even before you, and then the couple has this problem, like if there is a genetic abnormality, should we abort the child or not? And that is quite ethically confusing for many couples and even to doctors. So you are able to test the embryos before you implant them into the woman's uterus. And this was previously called as PGD or PGS. PGD being when you're doing it for a diagnosis. So say you have a family history of hemophilia or sickle cell anemia. You don't want your child to have it. So you can do a genetic analysis or a genetic diagnosis. So you're looking at specific diseases, right? Whereas in PGS or genetic screening, you're looking at all the 23 pairs of chromosomes to make sure that they are there or not, or whether there's any structural aneuploidies. So now you don't no longer call it PGD and PGS, but you call it PGT, pre-implantation genetic testing, and there are 3 types. PGTM, which is monogenic, for example, a single monogenetic disorders like cystic fibrosis. Aneuploidy testing is PGTA, and PGTSR is when you look at translocations and insertions. So just a small update. This is, if you consider it like a book of life, then your volumes 1 and 2 is what your genome is. The chapters are the chromosomes. The pages are the genes. You have the words as are the triplets. And then you have the letters A, T, C, G. Any error in this or any spelling mistake, you will have a genetic variant. And you also know about a chromosome. It has a short arm called the P arm and a long arm called the Q arm. And the DNA can either replicate and produce multiple copies, or it can be transcribed or transcripted to form RNA, which can later be translated to form a protein. And this is what the basic structure is. So what we are going to do in PGD or PGTA is basically looking at whether there has been any spelling mistake. So you have the egg and the sperm, you produce the embryo, then you take a few blastomeres and test whether it has a genetic error or not. If you have a genetic error, then that embryo gets discarded. If it doesn't, then you can implant it. So you have these options to prevent diseases. You have the options to check the embryo quality or the embryo genetic constitution in patients where there are a lot of repeated miscarriages. It's also used for social indication, for example, sex selection, family balancing. So if you have 3 boys in your family and you want a girl, then you can use PGT for sex selection. Or if you have 3 boys, then you need a girl, or then you can use it for female sex selection. It's not allowed in countries like Norway. So I don't think it's even allowed in India because we are not allowed to do sex selection. But in some countries, it's quite popular. You can diagnose a lot of single gene disorders, both X chromosome and autosomal inherited. You're basically looking at genes that can be faulty or inactive, and those embryos with those faulty genes are not transferred. You also have this thing about genetic screening where you're looking at aneuploidy. If you remember my last week's lecture where we talked about maternal age and its influence on aneuploidy. So over the age of 40, 58% of your embryos will be chromosomally abnormal. So it's important for maternal age. You can see that when I write AMA or advanced maternal age, it's written over 35 years. 35 is not a big age if you think about it, but from a reproductive point of view, if you remember the graph, the chances of pregnancy goes down a lot after 35 years. The chromosomal errors go up, and that results in an increased miscarriage. What are the techniques that are used? You use CGH, comparative genomic hybridization, and that is for analyzing the copy number variations relative to ploidy levels in both the test and the reference sample. You also use NGS, next generation sequencing, of the DNA and the RNA to study the genetic variations that are associated with a disease. We're using genetics to diagnose something. One example is a triplet repeat disorder. These triplets like adenine, thymine, cytosine, and guanine, they can be in different combinations. And if any error happens, or if the triplets are repeated more than the normal numbers, then you can have mostly a neurological disorders. Huntington's chorea is a typical example. Fragile X syndrome is also another example. I will explain to you one disease, Fragile X syndrome. It's an inherited intellectual disability. It's more common in boys than in girls. It's basically a gene called the FMR1 gene that's on the X chromosome. This gene codes for a protein that's very important in brain development and function. And when you have a faulty gene, then your intellectual ability is disabled. The repeat that it has is CGG, cytosine, guanine. Normally, if you repeat it from 6 to 50, that's normal. So some people can have 30, some can have 40, but then they're normal. But if you have between 50 and 200, it's a progressive disorder. It's called pre-mutation. It's a progressive disorder. You can have this intellectual ability decreasing after 50 years of age. If you are more than 200 in the male, then you have a Fragile X syndrome. If you are more than 200 in the female, they are unaffected carriers because they have 2 X chromosomes, right? So how is it inherited? So basically, if you have a Fragile X syndrome couple visiting an IVF clinic, and we intend to do a Pgta for them so that you make sure that the children don't carry this faulty gene. Say, for example, it's a mother who is a carrier. So she has X that has 1 X chromosome some of the X will have an X chromosome that is defective and 1 X chromosome that's normal. The father is not a carrier, so his X chromosome doesn't have a problem. So when they have children, there is a chance that one of the X chromosomes is the faulty one, goes into one of the girl child, because she has another X chromosome that is not affected, she will be a carrier. The girl, 25% of the girls will have no symptoms, but for one of the boys, it will be affected by the Fragile X syndrome because he has inherited the X chromosome, the faulty X chromosome from the mother. So because he can only have 1 X chromosome and the father is perfectly normal, even though it's only 1 X chromosome, he will still be affected. So when you're doing IVF, you're looking at the embryos, you're selecting the blastomeres or the cells to see whether this has a faulty X chromosome. And if you see that there is a faulty X chromosome, then you don't transfer them. So this is how the disease gets prevented. How do you do Pgta? The biopsy technique is very important. If you're looking at just female-related diseases, you can even start with the first polar body. The polar body is the product of meiosis that is being released. It has chromosomes, so you can test them. And if you see extra chromosomes in there, then you assume that the egg is okay. You can do it in blastomeres or cells. The cells of the embryo are called blastomeres. You can pick out 2 or 3 cells and do an analysis, or you can do it in a blastocyst where you get more than 100 cells can be removed. One of the advantages of doing a Pgta or a biopsy like this is that you have a lot number of cells for the blastocyst, right? So you have more information that is available. However, it's always interesting to know, to remember that human embryos have a lot of mosaic cells. So you can have some of the cells being mosaic, whereas the other cells being perfectly chromosomally normal. So imagine that the cell that you have taken out is mosaic, then you can make a false diagnosis. So that's one of the reasons that Pgta is still having the red, or Pgta is still having the red traffic signal. You can also use microarrays in Pgta. It's basically a platform for pre-implantation and prenatal diagnosis. You can use it to check complex disorders like, for example, diabetes, heart disease, cancer, and so on. But there has also been measures in some countries to use it as a general screening at birth, so you know that this child has the predisposition, say, for example, developing diabetes, so you can use it as a preventive treatment. So you have a test cell, you have a control cell, you do a whole genome amplification, then you do place it in microarrays. These are high order reproducible matrix of thousands of different DNA sequences that are attached to a solid one. So if you have a perfect mixture, then if you have both the copies, then it will be yellow. But if you have trisomy, that means that you have an extra chromosome, then the color shifts to red, right? Because this is the test cell. If you have a monosomy, or if you're missing one of the chromosomes, then it will be green because you have only the control cell there. So these can be used to detect aneuploidies. Another thing is Pgd with Hla matching, or what you might have probably seen the film or like read about savior siblings. These are siblings that are created by IVF that are used as saviors to treat their sisters or brothers. This book, The Miracles of Molly, it has been produced into a film. It explains the story of a young boy who had Fanconi anemia, and the sister was created so that her bone marrow could be used to treat this young boy. So HLA, or it's the human leukocyte antigen. It's basically typing. You have heard about HLA typing. It's different from ABO blood typing. So in this case, you're matching your intention is to match you with a donor for bone marrow or cord blood transplant. So this baby had an incurable genetic condition called Fanconi's anemia. You use HLA, it's a protein, it's a marker, it's present in almost all the cells of the body. Your immune system will use these HLA markers to know which cell is present in your body and which cell is not. That's the simplest way to say it. So the baby was created, or this baby girl was created, not having a Fanconi anemia gene or a defective one for that, and at the same time having an HLA marker that is matched to the brother. So once she was born, her cord cells or her cord blood placenta could be frozen, and then it could be given as a hematopoietic cell transplantation to the brother, and that's how the boy was cured of his anemia. So this has both its positive and negative. You're creating a savior sibling to treat another sibling that has a disease. It has its ethical problems, because why was this girl created? Was it created just to treat the sibling? So it's quite ethically controversial, but it has been used for treating a number of diseases in IVF. For example, hemophilia A and B, Huntington's disease, sickle cell anemia, and so on. So now we are moving from genetic diagnosis, time-lapse innovations, to the rapidly expanding field of uterus transplantations. In some women, they're born without a uterus. For example, in this Meyer-Rokitansky-Costa syndrome, or in some cases, the uterus will have to be removed, and then they have this chance of not having a baby. So the uterus transplantation is a rapidly expanding field. The first case of a uterus transplantation was in 2000, done in Saudi Arabia, where a 46-year-old hysterectomy, where she had to remove her uterus, it was transferred into a 26-year-old recipient, but it worked only for 99 days. In Turkey, it was transferred from a deceased donor to a recipient. That was the first case. Recently in Sweden, it has been transferred from a mother to a daughter, and the daughter in 2014 gave birth to a healthy baby. So in the case of uterus transplantation, you remove the whole uterus with the stalk, or what you call as a peduncle, and then transplant it into the recipient. And in this case, it was her own daughter, and she was able to give rise to a baby. So now you see, like when you don't have a uterus and you have to rely on surrogacy, you don't have to do that any longer if this becomes a norm. Then you have something called 3% babies. These are not designer babies, and this is your intention is not to create designer babies, but to prevent serious life-threatening diseases that are connected to the mitochondria. So there are a number of mitochondrial gene defect diseases, for example, Leigh syndrome. And mitochondria, is the powerhouse of our cell. When the mitochondria is defective, it affects the organs that use a lot of energy, for example, brain, heart, liver, muscles, and so on. In the UK, clinical studies have been approved in 2015, and the treatment has started in Newcastle, London, and Oxford. So this is a new branch. How is mitochondrial inheritance important? If you look at the B part of this one, this is how the mitochondrial DNA is inherited. This is the mother's egg, right? The mature egg. There's a nucleus. There are a lot of mitochondria. The mitochondrial inheritance in the embryo is only from the mother. So it's a maternal inheritance. In the father, the mitochondria is present in the neck or the midpiece, but during fertilization, that gets distracted. So the whole mitochondria of the baby comes from the mother. So if the father is affected, it doesn't really matter because none of the children will be affected as it's a maternal inheritance. Whereas if the mother is affected and it's completely like mitochondria, complete penetrance, then all the 3 children will be affected irrespective of whether they're girls or boys. If the mother is heteroplasmic, having a low level of penetrance, still the children will be affected. So your intention is to prevent the mitochondria from the mother, of the affected mother, to be transferred to the child. So how can that be done? The mother with the defective mitochondria undergoes IVF, the eggs are collected, the nucleus is removed, or the sick mitochondria, not the nucleus, I'm sorry, the sick mitochondria is removed. You use donor oocytes. You remove the nucleus from the donor because you have the nucleus from the mother, but then you add the mother's the nucleus into it, and then you fertilize it with the father's sperm. So in this case, you can say the genetics comes from the father and the mother or the same couple, whereas the mitochondria comes from the donor. So in that way, you can prevent the penetrance of the mitochondria. Of course, you cannot remove all the mitochondria, but you're trying to find out how much amount of mitochondria can be removed. So that's when to prevent mitochondrial disorders. You can also do mitochondrial transplantation, and this is called Augment Ivf. And the first baby was born in Canada, in Toronto, to a couple in 2015. What they did was they had several rounds of infertility treatments and nothing much happened because the mother's eggs were, even though she was young, her eggs were not having enough amount of mitochondria. So what they did was they used her own egg precursor cells and re-energized her own eggs by adding these mitochondria. I have to tell you that all these are now mostly in the experimental level, but quite interesting because if these turned out to can be used, then it will change the way IVF works now. You also have the whole branch of gene editing with the CRISPR and the Cas9 technology. The basic concept is find and replace function. You find the faulty gene, remove it, put in a guided molecule, and create a new strand. As simple as that. So in human gene editing, it has been used in human embryos. It has been used to treat Duchenne muscular dystrophy. So you cut the gene that is faulty, and then add the guided RNA so that you don't have the disease anymore. So it's like germline editing. It can be used to remove and then insert DNA. And this specific enzyme, Cas9, can cut the DNA at specific places. So how has been it used in human IVF? In China, in 2018, there was reports of the birth of twins where the CCR5 gene was removed. These are genes that actually can give you a predisposition to HIV. So the intention was that these 2 girls that were born will not develop HIV at all. And this gene-edited twins were born in China. It caused a lot of ruckus, mainly because when this scientist, he removed the gene or the CCR5 gene, he did not think about the fact that this CCR5 gene, even though it prevented HIV, it also could cause HIV. It also was important in a lot of other disease development, and it was not documented. So he ended up going to jail, and his whole study was removed. So it can be used in the mouse embryos. It has been used, or gene editing and this CRISPR and Cas9 technology has been used to develop healthy mouse embryos that did not have Duchenne muscular dystrophy. You can edit genes in eggs, sperms, zygotes, and embryos. The intention of doing this should be very clear. Is it for a disease prevention? So then it's widely welcomed. But if it's for genetic improvement and to develop designer babies, like how this Chinese scientist did, then it's quite questionable. There have been studies about cloning monkeys in the lab using IVF technology, using fetal fibroblast cells, and not done yet in humans, but if this technology is successful in other animals, then it could be the near future. So we were talking about sperms and eggs from the mother or from the donor. What about in vitro gametogenesis, which means that can you produce eggs and sperms in the laboratory? The first primate that was conceived with sperm cells from frozen pre-pubertal testicular tissue is Grady. He was born in Oregon. The sperm cells were artificially produced in the labs used from these pre-pubertal testicular tissue. You have this whole branch of in vitro gametogenesis, creating artificial eggs and sperms from embryonic stem cells. If you remember the blastocyst, I said that the ICM, or the inner cell mass, is a good source of human embryonic stem cells, or HESCS. And you can also, in this case, they were created either from the embryonic stem cells or from your skin cells in the lab. The epiblast and the primordial germ-like cells, when inserted into the female or transplanted into the ovaries, they resulted in eggs. And when placed into the testis, they resulted in sperms. So you can genetically, or like you can program these stem cells to create sperms and eggs, and they have given rise to viable embryos in mouse. How does this affect us? Because this is basically from your own skin cells. So you're not using third-party reproduction. If you remember last time, we were talking about third-party reproduction using donor oocytes, donor sperms, surrogate mothers. All that is gone. It sounds more like science fiction, but there have been studies that have successful experiments in mouse cells, and they have been conducting studies in human embryos, test embryos. The same thing again shown. This was from Japan in 2016, where they created lab-made sperms and lab-made eggs. And then they were able to show that all the three embryonic tissue layers of ectoderm, endoderm, and mesoderm were visible. And in the human eggs, it was first reported by The Guardian in the UK in 2018. The scientists took pre-pubertal egg cells from women, and of the 48 early stage eggs that they had, they were able to create 9 mature oocytes. So this is also being developed in the labs. But then you have also what's called as synthetic embryos, where you don't need eggs and sperms. They're artificially creating embryos or blastocysts. And this has been from the Netherlands. So you can see that when we were starting from sperm and eggs, and then we were using third-party reproduction of using donor eggs and sperms, now we have moved on to a stage where we are producing lab-grown sperms and eggs and blastocysts that we don't need us at all. So it's like synthetic embryos are being created. What about space age? Are we ready to move to the space age? So in 2017, this study came where the scientists at the International Space Station, they took a freeze-dried mouse sperm and stored it in the International Space Station for 9 months. And then they brought them back. Hello? And they brought them back to the Earth's surface to see whether the radiation effects are there. Because you can, in the International Space Station, the radiation levels are 100 times stronger than the Earth's surface. But when these freeze-dried mouse sperm stored there for 9 months were brought back to the Earth's surface, they resulted in healthy mouse pups. So we are ready to move to the space age. So I have given you a bit about what's actually happening now and what the future looks like. So things like time lapse, pre-implantation genetic diagnosis, artificial intelligence are already there, but things like lab-grown sperms and eggs, synthetic embryos are on its way. There are a lot of things in the middle, like for example, mitochondrial donations for preventing mitochondrial diseases. You have mitochondrial Augment Ivf, where mitochondria is regenerated and so on, that are quite experimental. So any technology that you have to introduce, you have to do the SWOT analysis, strength, weakness, opportunity, and threat analysis. And if you look at time lapse, the strengths are, of course, a lot. For example, you have uninterrupted culture, it improves embryo selection. But what about the weakness? You don't have so many randomized control trials yet, and it's quite expensive. So based on the SWOT analysis, you can really decide whether this new technology can be implemented or not. So basically, the 7 governance principles of your intention should be to promote well-being. The studies should be transparent. There should be due care. You should be doing responsible science. You should have a respect for persons, transnational cooperation, and finally, fairness in what you do. It's much easier to say that you can use your child's talent, designer babies, and so on. And maybe just when you do the ultrasound, you're no longer saying it's a girl or a boy, but you're going to say it's a smoker, it's a homosexual, an insurance agent, with 75% Alzheimer's probability. So you can really see how IVF has changed and how it's changing, and it's becoming a new branch. Thank you.
Now we have an interactive section. The participants can post your queries in the chat box, or you can unmute and ask your questions directly to the resource person.
What strategy is taken when multifetal pregnancy is encountered? It depends from country to country. In Norway, for example, or most of Scandinavia, you're not allowed to put back more than 1 embryo. So normally you don't have this factor of having a multifetal pregnancy. I know a multifetal reduction is done in many countries where you literally can, in a way, stop one of the embryos from developing further by doing a treatment. In Norway, that's not allowed. So we are not allowed to transfer more than 1 embryo. I think it's important to remember that the ultimate aim should be a single, or what they call as best. It's like the single healthy baby that's born. But multiple fetal reduction is quite often done. I didn't get, okay. How accurate is Pgta testing? It's not that accurate, because that's why it still has a red traffic signal. Why is it not that accurate? It's just because human embryos are mosaic, right? You can't take the whole embryo out and test. So even though you have CGH and NGS technology and microarrays, you might be taking embryo that is basically mosaic, or cells that are basically mosaic. And based on 1 or 2 cells, for example, if it's an 8-cell embryo, you can't take more than 2 cells. And just to remind you, like when you have an 8-cell embryo, it's totipotent, right? So if you take 1 or 2 cells out, it doesn't really matter. But if that cell is mosaic and you're testing that one and you're saying, oh, this embryo is not good, and you're throwing the whole embryo away. Whereas you have the other option where that cell that you tested is normal, whereas the rest of the cells can be mosaic. So it's not that accurate. Sometimes they say that if you have a Pgta testing, you have to, if you get pregnant, you have to repeat it with an amniocentesis or a CBS. I think, yes, okay. Will surrogacy or sperm donation affect the emotional bond between the baby and the couple? I think it's, I don't really know whether it will affect the emotional bond, but sperm donations, for example, here, or even egg donations are non-anonymous, which basically means that the child at the age of 15 has every right to find out who the donor is. I think it's quite important because now with all the ancestry testings and genetic testings that are available, you can get information. So openness, transparency is the most important thing, right? Surrogacy, I think for example, here, if you have a couple going to sperm donation or egg donation or surrogacy, surrogacy is not allowed in Norway, just to be clear. Sperm donation and egg donations, you go through this whole pre-evaluation before they even start thinking about the process. They have talks with the therapist, the psychologist, and so on. And both of them, and if the psychologist or the therapist gives a report that they're not ready, then they're not allowed to go forward. I don't think it affects the emotional bond because you can really say that a lot of adopted children are their own children. I have not seen, I've not seen or read articles where they affect the emotional bond, but of course it depends from person to person.
Ma'am,
Yes.
if there is a defect in the embryo observed through scanning, will testing the amniotic fluid harm the baby?
No. Amniocentesis, if it's done by a proper technician, it doesn't harm the baby. But of course, there's always a risk associated with any invasive techniques, right? So it is there, but it's not as much. I think somebody had raised their hands. I think it's important to remember because there's something about biotechnology, like you're working in the lab and you're doing this, but when you have it in a clinical setup, you have to remember that whatever you're going to do is going to affect the future generations, right? Because when you're working in an IVF lab, you're using sperm donations, egg donations, surrogacy. Sometimes we as healthcare providers forget the fact that on the other side of the spectrum is a child that's going to be born. So it's quite important that they're counseled very effectively. When you're paying money out of your pocket, it's quite easy to say, put back 2-3 embryos because I wouldn't mind having a twin, right? But all the side effects, all the health effects of having twin pregnancy on the mother, the preterm birth, now even like, preterm births have been linked to autism. So the best option will be to put back one embryo, have a single healthy baby, and make sure that baby is completely okay. And I don't think it really matters, but the emotional bond, there have been studies on children 5 years after they're born and 10 years after they were born using IVF, sperm donation, and egg donation, and they seems to be doing much better than normal children. And one of the reasons that was a big study from Scandinavia called CONARTS, CONARTS. So if anybody's interested, you should read about it. It's basically because they say that this is like a long-awaited child. So basically, the parents give more attention and care for the children. So that's one study. What do you think about thinking of this as a career for you, or is it something that's like too much, like it's too much of science fiction and stuff like that? What are your views about it?
It seems to be a more fascinating field, ma'am.
I think now we are seeing the boundaries between different sciences disappearing, right? Like you have biotechnology, your molecular biology, but then you have genetics added to it. And then you have artificial intelligence, which previously didn't have anything to do with IT, that didn't have anything much to do with science. You have diagnostics. So you have laboratory technologies added to it. So I think it is radiology, ultrasound. So I think it's an interesting field, or it's like a good marriage between different science fields. It's of course like very result oriented. If you're working in a clinical field, then if the clinic doesn't produce, if the center cannot guarantee, or I wouldn't say guarantee, but if they cannot help couples have babies, then it can be a stress, right? Because the doctors want pregnancies, the couple wants pregnancies, you want them to get pregnant. So it's like a roller coaster ride sometimes. But I think the end product of being helpful to somebody, being altruistic, because you should remember that sperm donations and egg donation, the people who donate them, they do it altruistically in countries like Norway. So they don't get paid a single money for this thing. So when they do it altruistically, I think it is also very rewarding in one way. So if you can, I would say, if you can, sometimes I don't get the English word because now I have to speak Norwegian, but if you can manage to overcome your stress levels, then I think it's a very good field.
Thank you, ma'am. With this, we have come to the end of the interactive session. Thank you so much, ma'am, for this informative session. We have got a lot of new information regarding the field, and it was really encouraging for a lot of us who are interested in this field. Now, I invite Dr. Lizzie Jacob, coordinator of this event, to speak a few words.
Am I audible?
Yes.
Yes.
Am I audible?
Yes.
Yes, ma'am.
Good morning, all of you. I would like to take a few minutes to thank our resource person, Ms. Shabana Said, for this very informative session happening for the last 2 Saturdays. On a personal note, she is my classmate during my PG days and a very dear friend. When I contacted her for this webinar, she was having a very tight schedule. She was actually having an audit from the health department. And in spite of all that, she consented to have 2 sessions on this ART. Thank you, Shabana, for being with us for these 2 sessions. It was 2 very simple but very informative sessions that was happening for these 2 Saturdays. I thank you for being with us, for sharing your knowledge, sharing your experience with our students. And we are looking forward for many more fruitful sessions with you. And on a personal, I would like to mention one more thing. One of our friends used to joke, like, Shabana is doing God's job. He used to joke, but apart from a joke, that's a reality. She is actually doing a very noble, a very divine mission. She's on a very divine mission. She's helping couples having reproductive issues have a child. So I pray that you continue with your mission, you reach greater heights in your career, and keep spreading the smile, happiness in many more families. Thank you for being with us. One more time, thanking you.
Thanks a lot, Liji. And we are not gods, but we try our best to help others. And I think that's what I said about being rewarding. You may not bring joy and happiness in everybody's life, but you feel that as long as you try your best and you give them a good service, I think it works. And I should say that when Liji asks me something, I cannot refuse. So hopefully I'll get to see some of you sometime. And if you have any questions or queries, or if you're interested in this field, then of course I will try my best to help you out. And thank you so much again to Liji and the rest of the biotechnology department for inviting me to give this lecture. I always like talking, so that helps. But I also think it's interesting that this is my passion and this is what I like to do. And it's always nice to convey it to others so that I feel that if one of you or the other is slightly interested, then always think of this like maybe a possible option for you to work. Thank you again.
Now, let me invite Jovita Roy to deliver the vote of thanks.
Am I audible?
Yes.
A warm good morning to all. Assisted reproductive technology can alleviate the burden of infertility on individuals and families. It involves a range of strategies to help people conceive. Its goal is the provision of safe, efficient, and affordable care to optimize the chances of having singleton pregnancies and the delivery of healthy babies. On this remarkable occasion, it's my privilege to propose the vote of thanks to all who have helped us in making this webinar a very ambiguous success. I, on behalf of the Department of Biotechnology and the entire fraternity of St. Berchmans College, first of all begin by giving glory to the Almighty who blessed us with today's valuable occasion. I extend my sincere gratitude to our distinguished speaker, Shabana ma'am, to take out time from her hectic schedule to grace this webinar with her motivating talk and presenting the topic, Innovations in ART, Impact on Contemporary Clinical Practice and Future Advances. Her patience and exceptional modesty in unfolding the subject indeed helped us in better comprehension of the matter. We are grateful to our head of the department, Mr. Shobin Verghese, for being a constant source of encouragement with his compassion, attention, and impeccable support. I must mention special gratitude to the coordinator of the entire program, Dr. Liji Jacob. Without her profound hard work and guidance, this occasion would not be possible. I am very much thankful to the entire faculty for rendering their valuable time and suggestions to improve the quality of the program. Finally, I thank all the wonderful participants for your enthusiasm and making the event an overwhelming success. An event of this dimension cannot happen overnight and needs meticulous planning and execution. Once again, I thank each and everyone for your successful involvement in this international webinar series and making it a pronounced success. Thank you.
The link for feedback form is in the chat box. All the participants are requested to fill up the form for issual of the e-certificates. Once again, thank you all and have a nice day.
Thank you. Bye.